Anyway, my name's David Harris. I'm the provost here at Tufts University. And I thank you for joining us today in general, but especially for this exciting session here as we come to an end on this very exciting series of events. I have to say, just on a personal note, this has been really exciting because I remember this is my 6th year at Tufts, and when I arrived, we started talking about this SEC, this complex, and what it could be, and talking with Tony about how it would help advance science at Tufts. I remember a few years ago when we first started talking about the Allen Center and the work with Mike, and to see all of this come together is just so exciting. It reminds us what's so important about Tufts, the special work we do, the special work higher ed does, and I just am pleased to be able to be a small part of this event today. We think of Tufts as a multifaceted campus. It's a university that values the humanities, that values the natural, applied sciences, the arts, social sciences, and just about everything else in between. We're thrilled that we're joined not only by many members of the Tufts community, but many members of the larger scientific community in Boston and around the country. The new SEC, which we are in part of it here, and hopefully had a chance to wander around and check it out, as I said, it was built to encourage collaborations across disciplines, across majors, and across the university. Thanks to the vision and support of the Paul Allen Frontiers Group, the Allen Discovery Center at Tufts was founded last year. Now, there are many big ideas out there, and thanks to the Allen Frontiers Group, your support is helping us to move forward on these big ideas, so thanks very much. Now let me introduce our panelists. So a really exciting panel here today. I'm excited to hear what they have to say. First, our moderator, Patrick Skarrett. Pat is a senior editor at STAT, which is a leading national publication reporting from the frontiers of science on compelling stories about health, medicine, and scientific discovery. He's the editor of First Opinion and works with experts from around the world to provide context and perspective for the day's news. He was previously executive editor for Harvard Health Publications, the consumer publishing division of Harvard Medical School. He's also worked as a high school science teacher and as a biologist studying fruit-eating bats. Pat earned a biology degree from Northwestern University and a master's degree in biology from WashU in St. Louis. We have 4 panelists with us this afternoon. Tom Skalak, who you heard from earlier today, joins us from the Paul G. Allen Frontiers Group. Tom's the founding executive director of the Frontiers Group, which seeks to explore new frontiers, reinvent fields in ways that reflect major societal challenges and fundamental scientific curiosity, and bring new knowledge to light with a broad array of partners, making a positive impact on the world. A major interest of the Frontiers Group is the wide and growing landscape of quantitative bioscience. Previously, Tom was vice president for research at the University of Virginia, where he led research and innovation programs spanning biosciences, environmental sustainability, physical sciences, engineering and technology, arts, design, and humanities. As a professor of biomedical engineering, Tom's personal research included biomechanics of the cardiovascular system, angiogenesis, computational modeling, systems biology, wound repair, and regenerative medicine. He's past president of both the American Institute of Medical and Biological Engineering and the Biomedical Engineering Society. Next up, we have our own Professor Michael Levin. Mike holds an endowed chair. He's professor in the bio department at Tufts and director of the Allen Discovery Center at Tufts University. Recent honors include the Scientist of Vision Award and the Distinguished Scholar Award. His group focuses on understanding the biophysical mechanisms that implement decision-making during complex pattern regulation and harnessing endogenous bioelectric dynamics toward rational control of growth and form. I do have to read some of these because I'm a sociologist. I also note that it's really exciting for students here. Mike was in this building a long time ago. Mike was an undergrad at Tufts, and it's really exciting to talk about a student-centered research university to see one of our own do so many amazing things in the world of science. Professor David Kaplan is next. David holds an endowed chair. He's professor and chair of the Department of Biomedical Engineering and also holds faculty appointments in the School of Medicine, the School of Dental Medicine, the Department of Chemistry, and the Department of Chemical and Biological Engineering. His research focus is on biopolymer engineering to understand structure-function relationships, with emphasis on studies related to self-assembly, biomaterials engineering, and functional tissue engineering in regenerative medicine. He serves on the editorial boards of numerous journals and is associate editor for the ACS journal Biomacromolecules. David also is one of the most collaborative people at Tufts University. As you can tell by all the places where he's affiliated, has a huge impact across the university and always has something really exciting and interesting to talk about. So I'm looking forward to the conversation today. And last, I'm pleased to introduce Professor Jessica Whited. Jessica is an assistant professor at Harvard Medical School and at Brigham and Women's Hospital. In her graduate studies at MIT, she pursued questions of pattern development and maintenance in the Drosophila nervous system in the laboratory of Dr. Paul Garrity. These investigations led Jessica to consider mechanisms that animals use to ensure body plans are preserved following injury, and she since worked to understand how salamanders regenerate entire limbs following amputation. Jessica's laboratory now works to combine her previous research with new gene expression data to elucidate molecular and cellular principles underlying vertebrate limb regeneration with the hopes that the knowledge will shed light on the more limited regenerative responses in humans. Sounds very exciting. Looking forward to it. So Patrick, I should take it away.
Thank you very much. Well, thank you for spending the last bit of today's symposium with us, and I hope it will help close out the day and get you thirsting for more knowledge, just as you'll be thirsting for the beverages at the end of this at this panel. So I'm sure you've heard or are familiar with Mike's work and the Allen Discovery Center at Tufts University for reading and writing the morphogenetic code, but you may not be as familiar with David and Jessica's work. So I think it would be great to take a few minutes for them to describe in broad brushstrokes their work. Jessica, if I can start with you, when was the first time you saw Ambystoma mexicanum, otherwise known as the axolotl?
Yeah, so the first time I saw it in person was actually when I showed up to work on them in the lab as a postdoctoral fellow in Cliff Taben's lab.
Was it
there were a couple
love at first sight?
Oh, yeah, of course, yeah. I was nervous about amputating their limbs because I had grown up working in invertebrates, so in fruit flies, which, you know, it feels different than it does to basically do an operation on a vertebrate, and so I was a little bit nervous about that, but I got over it quickly, and the rest is history.
Can you describe, for folks who don't know, these crazy animals?
Right, so these are neotenic salamanders, and that means they're permanently aquatic, so they don't go through that last part of the life cycle of a normal salamander. So they just live in the water, they have these big frilly gills, they can reproduce in this state, and they can regenerate their limbs throughout their entire lives. They can live to be 10 years old. And their limbs on the inside have all the same tissues as human limbs, so muscle, nerve, cartilage, bone, tendon, dermis, etc. And so what we really want to understand is how they can regenerate these limbs, because we think that there's going to be some really important clues in there as sort of a roadmap for developing hypotheses about how to go back into mammals in the future for therapeutic reasons someday.
Does a regenerated limb look regenerated, or does it?
It is a perfect replica, exactly the same size as the original one. In fact, you cannot tell which one was amputated because there's no scar on the outside whatsoever upon first amputation. But one of the things we're probing more deeply is how many times can you challenge the animal to regenerate the limb? And it turns out it's not infinite. And so our lab is now using this as a platform to discover the clues that might underlie this loss in ability to regenerate following repeated amputation.
So there must be a whole lot going on for them to get to do regeneration.
Right, so one challenge we have is that we don't have the full genome sequence of these animals yet, but we do have a really good proxy that we developed with the Broad Institute, so that would be the transcriptome or the expressed genes. And what they do after amputation is that they mount this regenerative response, and they heal the wound in a special kind of way that leads to the formation of this thing called a blastema. And that's sort of like where all the magic happens, where the progenitor cells go to proliferate and get repatterned and grow the new limb. And what we think is really key is that because mammals don't ordinarily respond to amputations by making blastemas, if we knew how blastemas form, we'd be a very large part of the way towards solving the problem, because they're required for regeneration and mammals usually don't make them. Yeah. So, yeah.
Thank you. If you'd like to see Jessica in action, I'm going to put in a little plug here, because one of my colleagues, Carl Zimmer, has a series on STAT called Science Happens. And one of the episodes is with Jessica, and by chance, the other is with David. So they're both featured. If you want to actually see their work in action, you can do that at STAT. So David, as I understand it, your work involves nerve cells and a very special substance, silk, which you don't ordinarily connect with nerve cells. What's the connection between nerve cells and silk?
So silk is a plentiful protein out there. You know it because you've worn it or slept on it. But it turns out to be a really remarkable protein-based biomaterial that you can morph into almost any form in terms of structure and function. And so as a lab that has sort of had its origins in how to engineer substrates to control cell and tissue responses, it turns out to be a really nice starting point. It's like a blank slate. It doesn't force cells in one direction or another. It gives them a chance to propagate and then provide other signals, whether it's bioelectric signals or biochemical signals, to do what you want. So it's a really nice substrate to grow cells and tissues in the laboratory, and when you're done, you can translate them directly in vivo because silk turns out to be very biocompatible. And you'll see a series of silk-based systems, materials, devices coming through the regulatory process for use in patients. So it's gratifying to see the expansion of the field over the last couple of decades, let's put it that way. And then at the same time, we spend a lot of effort to grow specific cell types towards given tissue outcomes. Usually that's structure and function. And one of our current focal areas is how to innervate human tissues in the laboratory on a lab bench so we can understand the signaling that goes on between nerves and other tissue types, whether it's epithelial layers, stromal layers, and the like, to really understand how the nerves are responding to those tissues when you perturb them, and then how this can lead to understanding of mechanisms of pain, sensation, regeneration, repair, and so on. And one of the areas, for example, Mike and I are currently exploring is how to compare nerve and vascular systems side by side, and what talks to what in regards to regeneration. So there's lots of important questions we can ask. We're fortunate to have some really talented postdocs, I don't know if they're still here, that are able to generate nerve stem cells from human cells that we use, as well as study these vascular nerve niches within tissue systems to lead towards new ways to think about regeneration.
One of the things that I saw, in Medicare drug coverage, the donut hole is a bad thing. For you, the donut hole actually is a good thing. Can you explain, how you're using the hole to answer questions?
Sounds odd, but food analogies always work for me. So one of the tissues we're very actively studying, and in fact will be part of this new complex here, is our three-dimensional human brain system. So think of a brain in a dish, and it's based on a donut. And the donut part is the silk spongy matrix. It turns out to have the right mechanics and the right sort of morphology to support neurons to continue their growth and function for long periods of time. And when I mean long periods of time in the lab, I'm talking a year, so we can really study things like neurodegenerative diseases, which are ongoing in the group. And in the center of the donut hole, we place a collagen gel. And for some of the students here who work with collagen, it's the bane of your existence, because when you make a collagen gel and you come back 7 days later, it's shrunk down to a little nub, and therefore you've killed all your cells. But it turns out the silk donut is a really nice holdfast for the collagen, so it can't shrink down. So you have an imaging window with nice collagen in the center where the axons generated by the neurons can form connectivity, and you can look at synapses and you can look at electrophysiology, and now we can start to perturb the system or use disease-related cells to study things like Alzheimer's and Parkinson's and autism, which are all very active areas of ongoing study in the group. And you can do this over months and years, the lifetime of a graduate student, right, to try and make some new discoveries about these systems. So that's where the donut design comes from.
Thank you. Mike, reading and writing the morphogenetic code sounds like it could be a Dan Brown follow-up. You've probably heard that before. What is the morphogenetic code, and what are some of the ways you're trying to uncover its secrets?
Yeah, so basically, we have the genome that everybody understands is important for patterning, But then in the end, you end up with an anatomy, a three-dimensional structure. So the genome, of course, doesn't encode directly for the structure. The genome has the parts list, you've got your proteins and some regulatory stuff. But in the end, there has to be a decoding that goes from what's in the genome through including the physical processes of development and regeneration, and eventually you end up with an anatomical structure or repairs to that anatomical structure. So the morphogenetic code is simply a way to understand how to read out and ultimately control those processes so that you can have a control over that three-dimensional structure. So you could affect repairs, make new structures, and so on. That's the code.
And Paul, a setup for you, Tom, sorry. The Paul G. Allen Frontiers Group, I think it's described its funding strategy as patient long-term investments in teams of scientists pursuing out-of-the-box approaches. How do you find them?
Good question. Well, we have the pleasure of looking everywhere. So we've told Paul Allen that we believe talent is everywhere. So that means we're on a continuous world listening tour, as we call it. So we invite ourselves to different universities, to centers, small ones, large ones, every geographical region. We talk to young people, we talk to established people, we listen to presidents and deans about their strategic plans, but we also listen to the youngest revolutionaries about what they think's not being done and where they might like to head into unknown territory, to discover the next valley over the currently visible peaks, if you will. So we're basically asking people what they think the dark matter of bioscience is, recognizing that it's a very young field. Bioscience now is on sort of a exponential growth curve in knowledge, and that knowledge will be the basis of many industries, of the future. Basically key to, winning the future of America for sure, as our former president liked to say, and also of the world, even extending to things like equitable uses of resources, even peace processes. So we think it's very fundamental, because when any field of knowledge is on that kind of curve, like chemistry and physics were 100 years ago, it has major implications for society. But to find them, you have to create the new knowledge. So we go and look to do that.
How many centers are there now?
There are now 4. So we named 2 last year and 2 this year.
Do you expect more?
Yeah, there will probably be sort of a string of pearls of Allen Discovery Centers over time. Again, recognizing that talent and ideas are everywhere. And we don't have a perfect crystal ball, so we don't always know when there will be a winning bet that's made. But we just use our best judgment. One way we think about it, however, actually is pretty well represented in this room. So we look to see who's listening. So in this case, Michael Levin and the team and center here is acting sort of like a center of force to stir up, interest in this idea of defining the morphogenetic code. And if I look in the room, I see Don Ingber, the founder of a Wyss Institute at Harvard, Nadia Rosenthal, who's leading probably the world's top place for building genetic animal models of disease and complex phenotype. You have Dean Kamen, probably the world's most iconic designer, inventor, sitting here, wanting to learn about the code so we can better design tissues to repair injuries and help people. John Abele, who founded Boston Scientific Corporation and pioneered less invasive medicine, changing the field of medicine completely. And when we see people like that are here to listen to what this team has to say, then we say, well, maybe that actually is a pioneering direction that others are following the expedition.
So listening to the listeners.
Yeah, it's continuous listening. Yeah.
In keeping with the dark matter theme, I'm going to share an insight from Dr. Charles Sidney Burwell, who was Dean of Harvard Medical School from 1939 to 1945. He was a cardiologist. He was dean when women were first admitted to Harvard Medical School on an equal footing with men. So that was kind of a turning point for the medical school. In an address to students, he once said, half of what we're going to teach you is right, and half of what we're going to teach you is wrong. Our problem was that we don't know which is which. So I'd like to ask all 4 of you, well, David, let's start with you. How do you go about illuminating what's right and nudging aside what's wrong? Any thoughts on that?
The adage is you learn more from what you do incorrectly than what you do correctly. So the simple answer is it doesn't matter. You learn, even mistakes are really good learning tools. And oftentimes that's what I obviously tell my students and my own daughter, that it's okay to make mistakes. That's how you learn, right? So that comes through parenting, it comes through teaching, and it comes through the research. I think too many graduate students think it's an easy path to get in there the first year or year or two, or in some cases 3, or some cases 4, and it's kind of treading water for a couple of years till you really have assimilated enough knowledge base from all the mistakes where you all of a sudden get that sort of key experiment going, and then the path becomes sort of very exponential at that point. So I think all of that is, what's wrong or what you do incorrectly or the mistakes you made. I think you just have to learn from them. And you sometimes don't recognize that they're wrong until you're past that point, and it's reflective at that point.
I mean, it's amazing to look back, and it's so easy to see what's wrong when you look back, but it must be hard when you're in the middle of it.
That's correct.
Jessica, how about you?
In my lab, I just really encourage the people in the lab to listen to the experiments and what the experiments are telling them, because I think otherwise, it's easy to see something that you think doesn't make sense in the current paradigm and ignore it, cast it aside. But if your experiment is good, if it's reproducible, if it's well controlled, then what it's telling you is meaningful. And there's already been a couple times in the lab where we found something that was surprising and went against the dogma. And luckily, I have people here who were really in tune with figuring it out, and so they kept working on it. But yeah, I would say listen to what the experiments are telling you, because we can't assume that we, like you say, know what the 50% that is not right is right this second.
Well, it's that listening to the dogma part. I mean, that if you look back at the history of science, so many people just continue with the dogma, and it's hard to give it the highway.
Yeah, I think it's hard to give it the highway, but I think sometimes there'll be an experiment that people think that you've stuck a fork in it. Like there's this, killer experiment, it's part of the canon, and it happened in 1938, and no one's revisited it since then. And once you go back and intersect it with your current findings, you'll realize that actually no one's touched it since then. We all just believed that was the end of the story. And not that the original experiment was wrong, but just that there was more than one way to interpret it, So.
Mike, how about you?
Well, I guess the first thing I would say is I think if 50% of what we think is right, I think that's probably optimistic. I think it'd be really cool if it was 50%. I'm not sure that's true. As far as how you tell the difference. So I don't know. I have a very sort of engineering attitude towards it, which is that I like anything that gets us to a new capability. If there's a way of thinking or a way to think about something that gets you to a new being able to get biological systems to do something in the lab that they otherwise either you couldn't do or that they don't normally do, then that's a beneficial way of thinking. And I think, to my mind, probably maybe this is where it came from, but actually one of Dan Dennett's books from years ago, the intentional stance, the idea that if you can profitably increase your prediction and understanding by treating a system as if it were, let's say, an information processing system, right? People always argue, say, you shouldn't think about cells as computational units. That can't be right. Well, I don't know if it can or it can't be right. What I know is that if you think of them that way, it lets you do certain things that you otherwise couldn't do, and that's good enough. I think we can't really know what's wrong and what's right, but we can know what's moving us forward to better or things you didn't know about before. And so that's what I do.
So looking for things that work.
Yeah, basically, yeah. Anything that moves you to the next better experiment that you wouldn't have done otherwise, that shows you a biological system in a way that you wouldn't have otherwise known, that's a good direction. I don't know if when you're into truth or not, but I know when you're in a good direction. And I think that's how you know you're in a good direction.
Oh, that's an interesting way to put it. Tom, any thoughts on that?
Well, that covered a lot. When we're looking for people that we think are pioneers, we look for a track record of creating new things, or as Mike said, kind of using a design sensibility. It's been said, design is the first sign of human intention, and so that's when you're creating things. Now, we all know nature and life has been created by millions of years of evolution. It's not the result of a human design, so it would be a little anthropocentric to look at everything that way. But I think a combination of those is necessary, because what we're all doing when we do experiments on living things or are trying to design solutions, therapies, or new living things like in synthetic biology, you basically are standing on a long line of experimental work that has given you a parts list, but it's worth abstracting, if you will. So we look for the habit of abstracting. I think when people say follow the data, it's just, follow what your experiments tell you. What human beings do, even if you're looking at a simple graph of data, you're always abstracting. When you go home, you think, how does this fit a pattern I've seen before? And if it doesn't, what could be a new concept or paradigm, which is a much maligned term, I think. It's actually a good way to look at the world, that would fit my observations. And if you have only the habit of fitting past patterns you've seen in your life, I think you're doomed to kind of obsolescence and to be entrained in the dogma, as you asked at the outset. But if you're always open to new designs and maybe creating new patterns or new theories or concepts that can explain your observations, then you're at least in a place where you're open to discovery and where you may be a more creative designer, depending on your view of the world. But I think it's sort of an attitudinal approach to life and to what you're doing professionally that is actually a critical element.
How do you realize when you're onto something new?
It keeps you up at night. You're thinking about it. Yeah.
Yeah, the other adage is don't do boring things. That's what we always say in the lab. Right? So.
I think another telltale I was just going to mention, since I agree with Michael that probably that estimate was way off. It's probably 10% is accurate or something like that right now in biology, just because you just look at papers, they get overturned every 6 months easily because we just don't know all the parts and how they work together. It's just where this science is right now. So I think another thing that you can often find by reading across the literature very widely, not deeply, but widely, you see jargon used many times in biology and medicine. And often if you're reading a concluding paragraph of a paper and you can't quite figure out what they're saying, but there's a lot of very large words, that's usually a sign that they're not very clear on what the mechanism or the concept is. And then you know you're onto something. So looking for jargon is another way.
What surprises, what things surprise you that we don't already know?
I can start. I'll give you just one. There are certain large categories that we're pursuing right now. One is immunity, because it's clear the immune system now lies underneath many diseases and processes we didn't think. Then systems approach, which we heard a lot about today from Jim Collins, for example. But the big one would be evolution, where many people would say one of the greatest scientific advances of the past 200 years is Charles Darwin's, survival of the fittest and evolution. But we really don't understand evolution very well at all. Yet if you go into any cancer center in the country and someone has a tiny spot of melanoma and you ask the experts, has this evolved since it first formed till when it become a dangerous tumor or metastasizing? They'll say, oh yeah, it's mutated and evolved at least 15 or 20 times. And then you ask, well, are you teaching that to your students or your graduate students in the clinic? Are you working on that with a pharma company? No, we're not working on evolution. So, and that's not exactly true because there are people working on this idea. Heard a little bit today about it. But it's not a central subject. And the reason has been that people don't want to go in the rainforest and curate a lot of species and compare species as Darwin did with the finches. However, now you can do rapid evolution experiments in the laboratory, and you can actually get at it. So much like this group is trying to look at the morphogenetic code, understanding the laws of evolution, if you will, or principles of evolution, I think would be very powerful, and it's very understudied if you look at the percentage of papers on that topic versus the whole genre of bioscience literature. It's a tiny fraction.
Do you think it's
Yet it's behind everything.
Well, I was going to say, is it applicable across many different?
Of course, all life emerged through evolution. We did. The trees outside the window, all the bacteria exchanging genes down in the gutter in the street below here. So it's a very important force for why things are the way they are. And when we look at designs to repair things, if you're not accounting for how they became what they are, then you're not using all the information that might be available. And I think it's been quite neglected.
Interesting. Anybody else? Things that, people would have thought we would have figured out by now or understood by now.
I'm by no means an expert on it, but I think sleep and like the relationship with like sleep and hibernation and what this has to do with like recharging cells in the body, or is it getting rid of garbage, or like what exactly is going on? What's the reason for it and what's happening every time it happens? I think that's fascinating and still misunderstood.
Yeah, I'll add. It's sort of the context of the whole discussions today, but I guess I'd call it the degree of maturation versus integration in the body. So I think of tissue engineering and regenerative medicine. It's really unknown still today. If you create a tissue in the laboratory and you want to implant it in vivo, what stage of development should that tissue be at to give you the best chance of success? And I think these are things Don talked about a little bit. It has to do with innervation, vascularization, but also the cells talking to each other. And we still don't know. You can take almost any tissue today, whether it's the cartilage or whether it's a heart tissue, and designing a patch or designing a plug, it's still a crapshoot. You just don't know where to mature that tissue to maximize the chance of success.
Yeah, for me, just as one example, I'm struck at still the number of things we don't know about the relationship between the genome and the body that is the subject of selection and under fitness. And so in thinking about this problem of planarian, how is it that they have a rock solid morphology despite this constant accumulation of mutations? Over the last few months, I've been talking to various people who study genetics and population genetics and the evolution of body plans and things like this. And I had a very simple question. How much, on average, how much mutation can a given species take before you expect to see a pattern change? Like how much, you're shooting bullets at the genome randomly, right? So obviously there's hotspots, but let's say just randomly you're shooting errors into it. How much of that can a certain morphology take before you expect to see anatomical changes? And we don't have a clue. Nobody has the foggiest notion of how to even begin to derive that, an answer to that. So this is like a super fundamental thing that we don't know how to estimate at all.
So to stick with surprises for a second, can you recall a moment when you were totally surprised by your research? Either a finding, something that you just could not believe from your own work.
Well, so recently we found that if you cut one arm off, the other arm also engages in a cell cycle activation response. And in fact, this happens throughout the body. And so I think I don't still know the answer about why this happens, but I think it's really interesting to think about that the initial response to amputation is not a local event at all. In fact, it's a systemic event that later gets localized to the part that needs to regenerate, and I think that's kind of cool.
Does it happen with all three other limbs, or
Yes.
the?
Yeah, from what we
Interesting.
can tell, and some other body parts too. And we haven't surveyed them all. It could be that there are intrinsic cells in each organ that are responsive, and so we don't know yet what happens to those cells, the fate of those cells. If the other arm doesn't grow bigger noticeably, but it would be interesting to see how that might get extinguished.
How did you find that?
Actually, one of the people responsible for it is in the audience right now. So what we found was, we were using the contralateral limb, the other limb, as the control, as people often do. And we noticed that that there was an unusual amount of proliferation over on that side, or cell cycle reentry in that side, compared to what we would expect for an animal that size and age. And so we just decided to take a stab at really just studying that per se and following that up to see if it was a real phenomenon. It absolutely was.
David, any surprises out of your work?
I would come back to our donut model that we talked about before for three-dimensional brain tissue. You go through months and months of trying to optimize the cocktail to get the nerves to be comfortable enough to want to make connections. And then, when the postdoc brings in the data and brings in the donut, and you see axons connecting and you see electric fields and so on working in the dish, is pretty exciting and somewhat unexpected, because you've tried so many times and failed, and yet you finally get the right mix of all those factors. So I think those kinds of exciting moments happen with every tissue we tend to try and grow, where all of a sudden the right combination gives you a functional outcome that you hadn't seen before. And then you're on your way to do all sorts of more applied studies.
Any holy moly moments for you?
Yeah, well, there's been lots, but I'll just talk about one. So one of the issues in this planarian field is that the data keep accumulating, but it's been really hard, and people have been at this for over 120 years, trying to come up with a model that really explains the results we get. Okay, functional. So you cut it this way, this happens. You put this drug on it, that happens. So we have a huge body of data on this, and so about 4 or 5 years ago, we decided to start on something we call the bioinformatics of shape. So really to try and create artificial intelligence tools to try to help human scientists to make sense of this data. So kind of like the way that people now can deal with sequences and sequence comparisons and genomes and so on, there's nothing really for shape. So we're trying to understand the functional data. So what we ended up doing, this was the work of Daniel Lobo in my group, who's now an assistant professor at the University of Maryland. So what we did was we came up with basically an evolutionary kind of machine learning system where what it does is it forms a bunch of hypotheses, follows them forward, checks them against the database. And we had to create this database first, which means going through every planarian paper ever and encoding it in this kind of special language. It took a long time. But basically this thing it evolves better and better hypotheses by randomly tweaking them, checking the outcomes against the database in a special simulator, and eventually the idea was that it would eventually find a good explanatory model. So a couple surprises. When I first suggested this idea, and I would go around and sort of talk about this at the end of my talk, I found out that biologists generally didn't actually believe in the power of evolution, because everybody basically said, you're telling me that we've been at this for how long, we don't have a good model, you're telling me that a bunch of random changes is going to forget it, that's never going to happen. I was kind of stupefied, but people fundamentally do not believe in the power of this thing to come up with novelty. That's one thing I learned. But then the aha moment for me was that not only did it find one, which turned out to be the first model in this field that was discovered by a non-human intelligence, not only did it find one, but the model it found was perfectly understandable. It was not this crazy hairball of like metabolism looking thing that nobody would be able to understand, even if it gave the right answers. That would be good, but not really satisfying. This thing was a small model, only 16 components to it. You could easily look at it, figure out what's going on. It found a perfectly good answer to the problem. And what it did was, it wasn't just number crunching. What it did was the most creative thing we do as scientists, try to come up with a model of what's going on. It wasn't just helping us crunch through large numbers of data. It did the part that we're the most proud of, which is having this insight about what is actually going on in this biological system. And this tool is able to help us even with that part. So that was kind of when that was one of the big aha moments for me.
Tom, how about you? You've spent years in research and foundations. Any, kind of eureka moment for you?
You mean from my own lab?
Yeah.
Yeah, that was a long time ago, but I have a few. I used to work on blood vessel growth, and the big problem is one way to think of it is why in adults, some people have, chest pain and they have a severe heart attack and other people don't. They can deal with it basically and adapt. And the reason is some people grow collaterals, like a river that gets clogged, and you grow collateral around the blockage. And some people, when you X-ray them or take an image of their heart, you see all these collaterals growing all over the place, and say, why is one different than the other? So we worked on that problem, and we wanted to know, where do the cells come from? The building blocks of these life-saving collateral vessels, small arterioles, basically. So we labeled them up with a label that lets you see dividing cells in the very smallest arterioles. The smallest ones are capillaries, the size of one red blood cell. And as they grow, they get bigger, they recruit these cells called smooth muscle cells, which are the muscular coating that lets an arteriole open and close to regulate blood flow, which is what they do. You need blood flow in your heart, you need blood flow in your gut, et cetera. And we discovered that the number of dividing cells locally was not enough to explain the growth of a collateral. So we didn't know where so it was a mystery. So really, when you find a mystery, is always the biggest aha for me. So we didn't know where they're coming from. And this was before people really knew a lot about stem cells or reprogramming stem cells. So there are only a couple of possibilities. They're coming from your bone marrow, and they're circulating and arriving at their location and participating in building that new channel, or else they had to be resident upstream in sort of a growth plate of a blood vessel. But there's nothing in the literature that a blood vessel has a growth plate like a bone has a growth plate where the growth comes from. And this still hasn't been resolved till now. So it's actually still a mystery where the cells come from that build collateral blood vessels, which is a life-saving thing. Happens in everybody, but we actually don't know the origin of the cells as of today, which is why I asked Nadia earlier about fibroblasts. You know, are there fibroblasts sitting around in your vasculature that are the building blocks? We actually don't know.
It would be
So
cool to
that-
be able to turn those
I would love
on
to
if
see that
needed.
mystery solved. Yeah.
Interesting. So one of the themes of, I think, your work is generation and regeneration. Science depends on regeneration in the form of new scientists who either come through STEM programs, you know, those are sort of the generated scientists, and the regenerated ones who come from other fields. What do you think are the biggest limitations for students in the biological sciences now?
I think it's not a great thing that people aren't required to continue in math and continue to learn to code. Like, I certainly hardly took any math in college, and I wasn't forced to, even though I was good at it, and wasn't part of a biology or a philosophy degree. And I thought things had changed until I asked the people in my lab to raise their hand if they knew how to code. And there were like 2 people in that room, and there were about 20 people because it was the summer. And so I think I'd like to see that change, because it is definitely a tool that will be obviously helping modern biologists. But as far as, you know, like hurdles go, I think there's a lot of anxiety about what happens after you get your degree. Because in the old days, people had this paradigm where you go off, you'll become a professor, and that sort of image is sort of fallen apart a little bit in the younger generation, and there's a lot of anxiety about funding, for instance. And even though there are lots of opportunities outside of academic science for people who get a PhD, there's not a lot still of information being conveyed about how to completely go about that. So those are 2 challenges.
I would just add, the earlier the better. You know, the earlier we can get students engaged in these topics and comfortable with math and science in the same way. But we're talking, you know, K through 12 as critical. So I think more and more programs that encourage that can only help.
Yeah, I would say for sure for students a background in computer science and not computational biology, and not just Java or whatever some people start with, but actual computer science where you learn to think that way, and the concepts. To me, that's critical. We have people graduate biology all the time never having seen those concepts at all, and I just think that it's crucial.
So you think that kind of thinking can inform other kinds of science?
Yeah,
or doing other kinds of science?
I do. Yeah, I think it's really valuable. Yeah.
That's a. Go ahead.
I would probably add data sciences, you know, beyond coding per se, you know, understanding very large data sets, which is becoming more and more important in biology. And I would say, you know, as a general rule, getting them involved early, absolutely, but with problem solving, I would say, you know, hands-on problem solving, because biology's history has been descriptive, and we need to become more predictive and design-oriented. So just learning how to build things, you know, take apart your bicycle when you're a kid, or, you know, if you're, you know, like bicycles, you know, drawing, painting, writing. I think, you know, we're losing some of those things as a habit in our earlier schooling years. So I think reading and writing are among the best training to be a good scientist, because it forces you to abstract, you know, observe the world clearly, abstract, make judgments and decisions, and then articulate what your new decisions or problem-solving approaches are. So that, I think those, along with hands-on problem solving, those are the 2 powerful things that people should do. You know, robotics competitions, you know, we have to solve a challenge or go do something to meet some specified challenge, you know, quantitatively and predictably is, it's fun, and kids do it naturally. We sort of drum it out of them as they go through their school year. So just keeping that alive through all the years of school will make a more powerful biologist when they hit college.
Tall order. I have 3 kids who went through the Boston Public Schools, and just to see the limited science education that they get is a challenge. Last question. Mike has a 17-year-old correspondent who posed a question to him, and it's this. If we alter the anatomy of the body, does the brain have to be changed also to take full advantage of the modification? I think that's a really cool question from anybody, let alone a 17-year-old. So anybody have, thought, it's kind of what comes first, and how does all this happen?
Operative word is full advantage. Operative words, because I think we would argue that the body could take advantage of that, but in order to gain the full advantage, you'd want to also upgrade the brain. Do you agree, Mike?
Yeah, I mean, the plasticity is pretty remarkable. We've moved eyes and nostrils and ectopic limbs and things like this. And generally speaking, the existing brain is pretty good at making use of whatever it's got. I agree with Jessica that maybe a full advantage, it would be useful to upgrade the brain as well. But I'm struck by the incredible plasticity of the brain already as it is. And I think we'll get pretty far even without having to do that. But maybe it's even better if we knew how to upgrade it.
But also what upgrade means, if you're just talking about rearranging and doing plasticity, then that's also sort of an upgrade, not necessarily adding more cells, adding new structures.
Yeah.
I would tell them that we should not view biology as a series of version numbers and upgrades like you get to your PC at home over the internet these days. So I think it's dangerous to use analogies from today's technical industries when describing biology. So it's a great question. It's because it could be divorced from that, but I'm suspecting it may come from that. Let me upgrade version 6.0 or 6.2. But if you're adding a whole new wing to an airplane, then you're going to have to control it properly, or else your airplane's going to probably be unstable and crash. So I think posed right, it's a good question, but it worries me a little bit that people think biology will be solved by versioning or by just more data. I think we actually need new ideas and new actual understanding, not just new versions of the current one.
So it's interesting to hear you talk about plasticity because your work sort of stands on the shoulders of the work of Carla Schatz and others who actually showed that the adult brain was plastic. So it's just making me wonder whose work 20 or 30 years from now is going to be the standing on the shoulders of your work. And I hope it's pretty cool. So thank you all.
Thanks, Pat. So I want to just a few things. One, I'm the bad guy because I have to tell you the conversation has to stop. So I apologize for that.
That's the end.
But I want to thank Pat for leading this fantastic panel conversation.
Thank you.
And thank all of our panelists for not only sharing with us throughout the day the amazing science, but also the really important conversation about how you do your science, about what excites you, and about these mysteries in the work. And I'm sure that in the conversation that occurs afterwards, there'll be interesting collaborations that occur and other mysteries, hopefully, that get solved. So the good part of my job is I get to tell you there's a reception in the atrium. And I hope you all join us down there to continue the conversation. Thank you.